So I just gave you in the beginning a small introduction about nucleic acids. Then I moved to explain you what was the difference between these three processes: replication, transcription, and translation, that we will study in more detail later. And I just finished talking about the different components of the nucleic acids. And now I'm going to move to talk about the structure of DNA and RNA molecules. To talk about the structure of the DNA, I just remind you that there were three key differences that I talk, and I want that you stop the video and try to write the three key differences. Try to think about what were the three key differences. Okay, let's see if you remember. Here there's one difference. Yes, one difference is that here there's a double chain. Here there's a single chain. Was one difference. The second difference is the type of nucleotides. Although they share these three nucleotides, both DNA and RNA, the uracil is unique, is exclusive to RNA, and thymine is exclusive to the DNA. And finally, the third difference is the type of sugar. So DNA has deoxyribose, and the RNA will have only ribose. So these are three different. So to summarize, RNA molecules exist only as a single polypeptide chain, while DNA molecules have two polynucleotides and they are spiraling around an imaginary axis, forming a double helix. Another feature that is important in the DNA is that the nucleotides, they pair up. So the nitrogenous bases in DNA will pair up and form hydrogen bonds. So here you can see the hydrogen bonds, and you're always going to see the guanine attaching to the cytosine and adenine always attaching to thymine at the DNA level. So maybe you can use some mnemonics. So you always remember that adenine attach always to thymine and guanine always attached to cytosine. In class we can practice some of these exercises. In my case, I use some mnemonics. So for example, to remember the complementarity between A and T, I just remember that in my computer there is a button that is Alt, and it reminds me that A and T have always to be together. And in my case, I also remember the Geico, the GNC, and this is a way that helped me to remember. You can use another one. So now let's try to practice some of them. So here you have your DNA template strand, and I want that you try to make the complementary. And remember, if you have an A, the complementary will be a T. So here you will start making a T. And if you have a T, you have 2. The complementary will be an A. So I want that you finish this, stop the video, and you can practice. So in the first one will be a T, then it will be an A, the G, remember Geico, C, T, and complementarity base pair is not only important in DNA molecules, in RNA molecule is very important. And actually there is a vaccine that is trying to explore how pairing the RNA to stick another RNA molecule to the RNA from the virus can destroy the virus. So let's talk about that. So generally between DNA, you always have a pair between A and T and G and C. However, in RNA, you are going to have a pair always between A and U. And so in RNA, thymine is going to be replaced by uracil. So you are going to have A and U pair. Okay, so this is something important that you need to remember and that we have talked before in this lecture. Another important thing that I told you is this, that in some cases RNA can fold themselves. So in this case you can pair up here and you can pair up here. You can create hydrogen bonds and they will pair up here. And the last thing that I told you is that, for example, in some cases, so you can have an RNA like this, and a way to regulate, to destroy the effect of this RNA is to have a complementary. And when they attach, this will not go from transcription to translation. And this is something that is found in our own DNA, in our own body. This is a common mechanism that has been explored in or model organisms like Drosophila. I have used this type of technique. And also, as I said before, they are exploring whether this can be used as a vaccine for the coronavirus. So if you have this COVID RNA here and you produce a complementary copy, they will tend to attach and there will be no infection. So to summarize that part, remember, complementary pairing can also occur between two RNA molecules, as I said here, or between parts of a molecule, as I show here. So this is the second important thing that you have to remember from this slide. To finish, I'm going to talk about something that is very important, is that you remember that DNA and proteins can be used as a tape measurement of evolution. So here we have three different organisms, and remember that the DNA is passed from generation, from your parents to your kids, and we can pass in generation through generation, and this can be used as a tool to determine which organisms are closer to each other. So for example, with the coronavirus, has been very useful to see how much the virus is mutating, and in that way you can track how to stop the virus. So, and how they do it? In a very simple way. You just have to line the DNA. So for example, this is the DNA of the macaque, this is the DNA of the chimpanzee, and this is the DNA of the human. In the case of the coronavirus, you will have COVID from different regions of the world. As the COVID nucleic acids are changing a lot, you can compare these things. And then you can see which one are similar and which one are different. So for example, here in yellow, all of these ones are similar. And however, for example, here you see that this region is not found in human and chimpanzees. So this can be a tool to tell you that maybe human and chimpanzee are closer together than the macaque. And comparing these bases, you can start finding how close or how far are some organisms. So just for you to remember, from this slide, I want that you remember that the linear sequence of nucleotides in DNA molecules are passed from parents to their offspring. And as a result, two closely related species are more similar in DNA than are more distantly related species. In other words, as human and chimpanzee are closely related, the DNA will look more similar to the one of the macaque. And if you have doubts or questions, I will be happy to answer in class. Have a good day. Bye.